Hello, everybody. So I'm Alexander Kell, and we continue our seminars. Today, our guest is Dr. Sudipto Das. Dr. Sudipto has got his PhD in cancer genetics, and he's a lecturer and a leader of epigenetic and gastrointestinal diseases research group within a School of Pharmacy and Biomolecular Sciences of Royal College of Surgeons in Ireland. His research is focused on understanding how specific epigenetic modifications impact development of progression of cancer, colon cancer especially, and inflammatory bowel diseases. Sudipto has published widely across many journals with high impact, including Nature Communications, Journal of Clinical Oncology, Cancer Discovery, and his group is funded by both national and international funding agencies, including Science Foundation of Ireland and European Crohn's and Colitis Organization. Today, Sudipto will present a talk about the latest achievement in understanding the role of master transcriptional regulators in gastrointestinal disorders, especially. That will be a very interesting talk, I know. And after his talk, he will be happy to answer your questions that you can write in the Q&A chat in Zoom. And please don't hesitate to write your questions. They will be answered. And if not live, then we will answer them also written. So Sudipto, the floor is yours.
Thank you very much, Alexander. I'm just going to share.
So I think you have to switch on the microphone.
My microphone is switched on. Can you not hear me?
Yes, now we can hear you.
Okay. I'm just going to share my screen so you'll be able to see the slides. Okay. I'm presuming you can all see the screen?
Absolutely. Everything is fine.
Perfect. Okay, great. So again, thanks a million, Alexander, and geneXplain team in general for giving me this very kind invitation to speak today and for the lovely introduction as well. So as Alexander mentioned, I lead the Epigenetics of Gastrointestinal Diseases Research Group at the Royal College of Surgeons. We are based in Ireland, in Dublin. That's the front part of our building there that you can see. And what I'll try and do today really is to try and highlight really what we have been doing in the group, particularly using the geneXplain platform and focusing on master transcriptional regulator mediated signaling. And within that, what I'm going to try and do is kind of give you a bit of a glimpse on three different stories. One that we recently published earlier this year, and another story that's in review, and finally another kind of a study that's in progress at the minute. So I'll start, I suppose, with talking about what we do in our research group. So our group really kind of focuses on two distinctive gastrointestinal diseases that we've been working on over the last couple of years: inflammatory bowel disease, and within that we're looking at ulcerative colitis and colorectal cancer as well from the cancer aspect. We are really focused on looking at multi-omic profiling. So this includes, particularly obviously given our specialty is epigenomic profiling, but we've done a significant amount of whole genome and transcriptomic profiling as well. And with the idea is that we combine these different data sets together, particularly using both standard, gold standard bioinformatic approaches, all the way to deep neural network or machine learning based approaches, to combine this multi-omic data sets with phenotypic data. And once we've identified our targets, we ultimately kind of try and look towards validating these targets functionally using a number of different models. So we've got two different kinds of models that we have developed. Ones are organoid-based models, and the other ones are 3D bioprinted models. And within these particular models have actually also given us a really good insight or an opportunity rather to screen a number of different epigenetic targeting agents that we can screen using these models of both IBD and colorectal cancer and evaluate a number of different kinds of phenotypes. So to begin with, I'll start with the very first study that I wanted to kind of show all of you is focused on colorectal cancer. So as most of you would probably know, that colorectal cancer incidences are significantly higher. In fact, it is the most colorectal cancer associated deaths account for the largest cancer associated deaths globally if you discount gender-specific cancer types like prostate or breast cancer. But what's interesting from an epidemiological perspective really is that colorectal cancer rates have been significantly higher in the first world countries. And as you can see over here, the incidence are particularly high in the Americas, both North and South, as well as in developed countries within Europe, as well as across Australia as well. So in general, in terms of the etiology of the disease really starts presenting itself in quite a sequential format, mostly going from an adenoma to a carcinoma sequence, where at stage 0, where you see an incidence of a polyp, which then develops into a primary tumor, an adenoma, and that then grows into a carcinoma, which ultimately then disseminates into different organs. And this is basically given from stage 0 all the way to stage 4, which essentially is the metastatic colorectal cancer subtype. If we look at the National Cancer Registry of Ireland, this is actually quite interesting. And if we were to focus on the level of mortality in the patients, you can see that the rate of survival, particularly in stage 1, 2, and 3 patients, is significantly higher, all the way from 67 to 95%, whereas metastatic colorectal cancer patients, the survival rates is as low as 12%. So within the context of colorectal cancer, one of the first questions we wanted to ask was, we obviously know that there is, apart from stage, there is also disparity in terms of response to treatment, as well as the fact that patients of different ethnic or ancestry backgrounds actually respond quite differently to treatments and also have quite a distinctive prognostic rate. Apologies for that. PowerPoint just seems to have crashed. If you just give me a second. Okay, hopefully this works now. So in the context of this particular question that we were interested in looking at, one of our main kind of focuses was really on looking at the role of master regulators that impact cellular disparity in patients with colorectal cancer. And this study essentially was led by Dr. Parvati Meyer, who's a gastroenterologist based at the Albert Einstein College of Medicine in New York. So the rationale for this study really is from the fact that if we look at any kind of cohort-based studies, particularly in colorectal cancer, we know that apart from having a disparity in gender, say for instance, and age groups as well, what we do know is that these patients come from different ancestry backgrounds. And what is particularly interesting from a colorectal cancer perspective is that colon cancer patients who come from an African ancestry, in fact, have a poorer prognosis compared to colorectal cancer patients from European ancestry. So the question that we really had in mind was, what is driving this sort of a differential prognostic rate, and what is the underpinning cellular phenotype for these patients? To do that, we essentially looked at the Cancer Genome Atlas, which, as most of you would know, is one of the most comprehensive compendiums which encapsulates a number of different multi-omic data types, including DNA methylation, transcriptomic data sets, as well as whole exome. And looking at this further, one of the first questions we asked was, what genes are turned on or turned off between these two different cancer types? Is there a difference in anti-cancer immunity, given the fact that there is a difference in prognosis? Is there a difference in their immune landscape between these patients? And finally, do epigenetic changes in DNA, such as DNA methylations, ultimately differ between these two ancestry groups? So the very first thing that we really did was a principal component analysis that you can see over here, where we essentially predicted the ancestry of these patients in total. And here you can see quite clearly in general that the Caucasian patients separated out quite nicely from the patients colorectal cancer patients of African ancestry. To describe the cohort a bit further, what we essentially, we started with a total of 636 colorectal cancer patients who came from different ancestries following our prediction of ancestry. Now, the problem here was that not for all the European ancestry patients or African ancestry patients did we have usable multi-omic data. So what this essentially the usable data that we had was 55 microsatellite stable or MSS African patients of African ancestry, and microsatellite stable European ancestry, where the numbers were about 456 patients. So the very first thing that we looked at was looking at the actual tumor mutation burden, looking at using the whole exome sequencing data. And here we didn't really see a significant difference in the tumor mutation burden between the African and the European ancestry patients. Following on from there, we looked at the most commonly mutated genes across both the African and the European patients. And while you don't really see a significant difference in them, we do see that the hierarchy or the actual propensity in which these genes are mutated seems to be somewhat different. So say, for instance, if TP53 in KRAS are the second and the third most mutated genes in the African populations, it is in fact TTN and P53 that predominates this particular landscape in the European patients. We have recently, however, overcome this challenge, and this is predominantly owing to the lower patient numbers, where we have now recently published earlier this month a paper in Cancer Discovery that describes the overall genomic landscape of colorectal cancer patients across both these ancestry patients, across thousands of patients that have been sequenced through Foundation Medicine. So the next thing that we did was we started looking at the gene expression alterations between these two groups, and we did identify significant number of genes that were differentially either upregulated or downregulated in the African patients compared to the European patients, as you can see from this volcano plot. We then carried out gene ontology analysis, and here I'm just showing you genes that are actually downregulated, particularly in the African patients. And what was interesting for us to see was number of different pathways, and predominantly immune-associated or inflammatory pathways seems to be enriched in the genes that are downregulated in the African ancestry patients. More importantly, when we carried out a deconvolution using this RNA sequencing data, what we see is that the cytotoxic T lymphocytes are significantly reduced in African patients, and similarly the neutrophil populations are significantly reduced in the African patients when compared to the European ancestry patients. So ultimately what we do see is that largely these immune-associated genes are being dysregulated in the African patients when compared to European patients with colorectal cancer. The next thing for us really was to try and understand what was the kind of overall composition and to identify the clinical covariates within this data set. So for this, we carried out a univariate analysis across the entire cohort. And what was interesting for us to see is that while the African patients were mostly young, and then you can see that as from the p-value, but more interestingly, we also see that African patients, in fact, did tend to be either obese in class 1 or class 2. And similarly, from a tumor sidedness as well, we see that a large proportion of these patients, in fact, the African patients, seem to have right-sided colorectal cancer. So bearing this in mind, we wanted to kind of look at tumor side, ethnicity, and age as the three key covariates. And what you can see over here is that cytotoxic T lymphocytes consistently are significantly reduced in the African ancestry patients compared to the European ancestry patients. But more importantly as well, we do see certain association of the cytotoxic T lymphocyte with the sidedness, for instance. And this was kind of surprising to us because what you can see over here from this particular graph is that the left-sided tumors in fact tended to have lower cytotoxic T lymphocyte. This is actually quite contrary to literature, because in literature the immune infiltration in fact is much higher in the left-sided tumors compared to the right-sided tumors. And similarly in the stage as well, you can quite clearly see that in the African patients consistently, you do see a significant reduction of cytotoxic T lymphocyte, and there is also a difference in terms of stage. We see quite a similar patterns when it comes to neutrophils as well, that you also see the similar patterns of neutrophils being significantly reduced in the African ancestry patients, and similarly in the sidedness as well, that the left-sided tumors tend to have lower amounts of neutrophils compared to the right-sided tumors. So our key question then was what essentially was mediating these kind of changes. And to do that, we started looking at master regulators. Now, just to give you an idea, and I'm pretty sure most of you would know about this, master regulators are key regulatory molecules that tend to sit on the hierarchy, within the hierarchy of signaling. They tend to sit on the top of this hierarchical network. And these master regulators, what is known about them is that they signal through intermediate signaling molecules, followed by signaling through transcription factors, which ultimately causes up or down regulation of genes. So what we essentially did was we took our list of up and down regulated genes and we applied this to the geneXplain platform. And one of the first things the geneXplain platform does is carry out an upstream analysis where it takes these genes and identifies the transcription factor binding sites using the transfag database. Following from this, once it has identified these transcription factor binding sites, it then alludes to identify the intermediate signaling molecules, followed by the master regulators, ultimately developing a clear picture in terms of how these genes are ultimately regulated through this complex networks of master regulators mediated by transcription factors. So what we did identify obviously here was distinctive transcription factor binding sites in the downregulated genes in the African patients, in downregulated genes in the colorectal cancer patients of African ancestries. And we see the similar sort of a thing happening in upregulated genes as well. But what was really interesting for us to see is that majority of these master transcription regulators that actually seem to be downregulating genes in the African ancestry patients, most of them have an immune nature. For example, IL-8, CXCR1, CAP18, granzyme B, GM-CSF, RANTase, all of these have a significant immune or inflammatory role in general. The other thing as well that we also see in the case of upregulatory, we in fact, as opposed to seeing a number of different master regulators controlling downregulated genes in African ancestry patients, we identified DBL and GP6 to be the key master regulators regulating genes that are upregulated in African ancestry patients. So we then essentially asked the question, looking at these different master regulators that are downregulated and the transcription factor bind and the transcription factors as well, is there a correlation between cytotoxic T lymphocytes and neutrophils, as we have observed in across the entire through our data that we had to date? So when we started looking at this, what you can see from these graphs really is that we see a significant correlation of RANTase, CCR2, IFN gamma, PDG6, and GR across both African and European ancestry patients when it comes to cytotoxic T lymphocytes. And we see the very similar sort of a pattern for CXCR1, IL-8, IL-1 beta, CCR2, and SLUG that seems to have a significant positive correlation with neutrophils. And what this essentially suggested to us was that it is quite likely that these master regulators ultimately mediate the low levels of neutrophils and cytotoxic T lymphocytes in these colorectal cancer patients, which ultimately leads to this immune exhaustion sort of a phenotype. Now, the other interesting factor for us really was that one of the transcription factors that we did identify from our study was a transcription factor called BCL6. Now, BCL6 is quite an interesting transcription factor in that it actually has been associated with driving different inflammatory, quite a distinctive inflammatory phenotype, particularly in the. And in this instance, what they have really shown, or what literature really points out, is that we do see a distinctive, we do see BCL6 essentially driving particularly, driving this particular activity through IL-6 and GATA3 and macrophages and T regulatory cells. So given the fact that we do see BCL6 coming up as one of the transcription factors through which IL-8, IL-1 beta, CXCR1 and CXCL8 seems to be mediating its function, further substantiated to us that BCL6 ultimately might be a key regulatory molecule that impacts this immune exhaustion phenotype in the African ancestry patients. So we next moved on to looking at the epigenetic landscape of these patients. So we looked at DNA methylation, particularly from these African and European ancestry patients. And what we see was that we get a number of different differentially methylated sites between African ancestry and European ancestry patients. And most of these differentially methylated sites in the African ancestry patients appear to be hypomethylated, or which will tend to lose their methylation when compared to European ancestry patients. And looking at the different regulatory regions that these methylation sites are kind of interspersed, majority of them appear to be present in promoter regions of genes or within the gene body. So when we kind of combine this DNA methylation and gene expression data sets together, we identified 5 key genes that seems to have an association between methylation and expression. And what was really interesting within this was a particular gene called PPBP, which the other name for PPBP is CXCL7. And what is known quite widely in the case of CXCL7 is that the CXCL7, CXCR1 and 2 axis is a key driver of growth in renal cell carcinoma, More importantly, CXCL7 has also been associated with poor prognosis in colorectal cancer as well. And we particularly see that CXCL7, in fact, is hypomethylated and upregulated in the African patients compared to the European ancestry patients. Similarly, we also see another gene called LRRN1, which again has poor prognosis in terms of gastric cancer as well. So ultimately, what this suggests to us that we do see methylation-sensitive loci which have disease association not just within colorectal cancer, but across other GI disorders, but more importantly as well, that these methylation-sensitive loci might also impact the immune-associated differences that we had observed between African and European ancestry patients. So based on the results that we really had, we kind of postulated this mechanism, essentially suggesting that in colorectal cancer patients of African ancestry versus European ancestry, we see this reduction in anti-tumor immunity, highlighted by the lower levels of cytotoxic T lymphocytes and neutrophils in the African ancestry patients. Underpinning this anti-tumor immunity difference between these two patients lies master transcriptional regulators like CXCR1, IFN gamma, IL-1 beta, RANTES, which ultimately controls number of different transcription factors, which either controls upregulation that you can see with these green arrows, or downregulation that you can see with these blocked red arrows, which ultimately impacts this anti-tumor immunity. So the next study that I want to kind of share with all of you is a study that we have recently submitted for publication. And this study really looks at the role of master transcriptional regulators in driving the molecular changes that are mediated by new therapeutic modalities. And this work essentially is led by Ellen Finnegan, who is a fourth-year PhD student in my group. So the study essentially kind of centers around a quite an age-old problem associated with histone deacylase inhibitors, which tend to have a limitation in solid tumors. And that's why they have the treatment in solid tumors is actually quite limited. These HDAC inhibitors, like Belinostat, say for example, is quite routinely used in treatment of relapsed and refractory T-cell lymphoma. And the reason why their usage has been so limited in solid tumor is because HDAC inhibitors like Belinostat contain this hydroxymic acid moiety. When the drug enters the tumor microenvironment, because of the redox environment of the cells, this drug essentially, or this hydroxamide group, essentially undergoes rapid or premature metabolic degradation, ultimately making the drug useless. So what we essentially did was we collaborated with our medicinal chemist within RCSI, and they developed this complex compound, which essentially complex Belinostat, and you can see the hydroxamide molecule over here, with copper chloride. And this molecule, which we call as Copper-Bis-Belinostat, essentially contains this copper ring, as you can see here in the middle, and two molecules of Belinostats on either side. Now, this drug has been created with the rationale is that when the drug enters the cell, the Cu2 plus will get reduced to Cu plus, ultimately dissociating from the Belinostat molecule, but in parallel protecting this hydroxamide molecule from premature degradation and ultimately increasing its anti-tumor efficacy. So one of the first things that we did was we evaluated the compound Belinostat and its novel derivative that we had derived, Copper-Bis-Belinostat, across 3 different colorectal cancer cell lines, SW480s, 620s, and KCO2s. And what was very interesting for us really was that the IC50, or the inhibitory concentration 50, that kills 50% of the cells, what we see here in the case of SW480s is that for Copper-Bis-Belinostat, the IC50 is in fact half compared to Belinostat. And what this means is that you in fact require half the amount of drug to kill 50% of the cells in terms of our newly developed molecule compared to its parent form. But more importantly, this IC50 in SW620s and KCO2s remain quite similar or quite identical in general. So using these IC50s, we next kind of ask the question, okay, we have now done this complexation. Does this in fact really prevent the drug from undergoing premature degradation? So what is known about Belinostat is when that enters the body, one of the main sites of metabolism of this particular drug is in the liver microsomes. So there is a particular enzyme called UGT, which stands for uracil glucuronyl transferase. And UGT essentially breaks down Belinostat to its active its metabolite called Belinostat glucuronide. So what we essentially did, we did an in vitro experiment where we incubated the microsome with both belinostat and copper-bis-belinostat across a range of different time points. And what was very interesting for us to see is that for copper-bis-belinostat, in fact, the rate cells and the intrinsic rate of belinostat from the microsomes is reduced by 2.5-fold after treatment with copper-bis-belinostat, suggesting that this complexation ultimately is sort of preventing this drug from undergoing metabolic degradation. And this was carried out using mass spec, where we essentially tried to look at the level of belinostat remaining by measuring the belinostat glucuronide metabolite. So moving on from there, the next kind of thing that we wanted to look at was, after we've developed this particular compound, does complexation of belinostat to copper in any way, shape, or form inhibit or kind of, sort of impede its HDAC inhibitory activity? So to do this, we essentially looked at, again, the 3 cell lines using an ELISA-based assay. And what you can see here is that consistently, both belinostat and copper-bis-belinostat maintain their HDAC inhibitory activity, where while we don't see a significant difference in this HDAC inhibition activity between both these drugs, in certain instances like the SW620s, you do see a modest, probably a higher HDAC inhibition, which is at a moderate level in copper-bis-belinostat compared to belinostat. But the point here really was that complexation of belinostat with copper chloride did not impact the actual endogenous HDAC inhibitory activity of this drug. So we next try to validate try to examine the impact of both copper-bis-belinostat and belinostat and compare these two drugs across different phenotypes. To do that, we first looked at the cell cycle. And what you can see here is that one of the most striking difference that we see is that for KCO2 cells, when the cells are treated with copper-bis-belinostat, you see a decrease in S-phase population. But most importantly, what we do see is that the cell lines seem to have a very kind of distinctive phenotype when it actually comes to cell cycle arrest, which suggests that there might be an individual, this could be because of the individual integrity of the underlying cell cycle checkpoints. We then looked at apoptosis in general, and while both the drugs, belinostat and copper-bis-belinostat, consistently mediate apoptosis across the 3 cell lines, we don't really see a significant difference between both these drugs. So what this really suggests to us that copper-bis-belinostat in KCO2s is likely acting as a cytotoxic and a cytostatic agent, whereas in SW480s and 620s, this likely seems to be more of a cytotoxic effect. So the next question that for us was that is there a gene expression difference that these drugs actually underpin? So without going into too much detail and just kind of showing you the differentially expressed genes that were unique to copper-bis-belinostat, so we carried out RNA sequencing analysis post IC50 treatment of these, following IC50 treatment across all these 3 cell lines. And while we don't see a significant difference in gene expression profiles between belinostat and copper-bis-belinostat in KCO2 cell lines, you do see number of genes being different in the SW480s that you can see over here. And there were multiple genes that were upregulated uniquely by copper-bis-belinostat, whereas in the case of SW620, you only see ZNF114 and CD22 to be uniquely downregulated. So when we look at the SW6 at the top pathways that are significantly downregulated by SW620, you see a number of different pathways that come up, particularly in the case of Rho protein signal transduction and various immune-related pathways as well that seem to be downregulated by SW620. So the next question for us, in line with our previous studies, was to understand what is controlling this particular differential gene expression profile between these two drugs across both cell lines. So what we saw was that there were a number of different, firstly, between both the drugs, you do observe certain common master transcriptional regulators that appear to be regulating gene expression across the board through the similar sort of transcription factors, as you can see over here in this network. But more importantly, if you start looking at the master transcription regulators of genes that are downregulated by belinostat, you see SRC and LCK. And similarly, in the context of copper-bis-belinostat, and this is particularly in the case of SW480 cells, we see that the genes that are actually downregulated by copper-bis-belinostat seems to be regulated by VEGF, LCK, and ERBB2, which is also called as HER2 commonly. We then looked at how these particular master transcriptional regulators themselves correlate between control, which was DMSO treatment, compared to belinostat and copper-bis-belinostat in both 480s and 620s. And what we can quite nicely see here is that there is a significant difference in these master transcription regulators. However, these master transcription regulators seems to be more significantly expressed in the case of copper-bis-belinostat that you can see here from this green box plots compared to belinostat, suggesting that it is quite likely that this signaling that we're seeing unique to copper-bis-belinostat in SW480 cell lines is mediated by ERBB2, VEGF, and LCK. So based on this particular, on our observations, what we really could postulate is that this complexation of belinostat to copper chloride ultimately increases the in vitro drug half-life that we observe through our in vitro microsome-based experiment. And what we know is that once the drug enters the cell, the Cu2 plus gets reduced to Cu plus, ultimately releasing belinostat, protecting its hydroxamide group from premature degradation. This belinostat molecule then inhibits HDAC inhibitory activity, and ultimately what we know is that this induces apoptosis. And what we understand from this is that while the impact is cytostatic or cytotoxic dependent upon the cell line, largely number of different gene expression changes mediated by master regulators like VEGF-A, ERBB2, and LCK ultimately underpins this differential gene expression response which potentially might drive the observed phenotype that we see between these two drugs. So with that, I'd like to thank a number of people, particularly people from my own group, obviously Ellen and Lola, who I mentioned before. We also work quite closely with Professor Darren O'Connor's group as well in RCSI, all our collaborators, both industry and academic partners, our funding agents, particularly SFI and the Irish Research Council, and all of you for your attention. And just before I say a final thank you, just to do a bit of a shameless promotion, is that we do have a postdoctoral position available in our group that has gone live now. So if you know anybody, please do circulate the word out. I'll be very happy to chat to anybody offline. So thank you very much, and happy to take any questions.
Thank you very much, Sudipto. So that was really interesting talk. And that was a great, talk. Actually, I learned a lot. Let's have a look first to the questions. Yeah, it's we have one interesting question. I will read it for you. So, sure. What is the gut biome association?
So the gut biome association, there are recent studies that have actually shown dysbiosis in the gut, particularly in ulcerative colitis and colorectal cancer patients as well. I think one of the recent studies that I came across a couple of weeks ago was that Fusobacterium seems to be playing quite an important role in terms of IBD. So these studies are kind of becoming more and more clearer in that shows significant kind of differences in patients who progress versus patients who don't progress in terms of how their gut dysbiosis is as well. We don't see as such associations in our own study because obviously our samples are either biopsies from patients or they would be available data already. So we don't have, like, for the likes of doing microbiome analysis, you'd really need the stool sample essentially to be able to do the microbiome analysis. So we haven't really looked into that aspect yet. But majority of the things, in fact, the inflammatory process that I've shown as well, but what is known as is that these inflammatory processes mediated by the master transcription regulators could also be mediated by a change in a change in the kind of bacteria that is enriched in a patient.
Mm-hmm. Very interesting. Yeah, right. Actually, until next question will come. So just to follow this question, sure. It's actually right. So the microbiome seem to be playing a really great big role in gut inflammation. And in case of IBD, what is the status concerning the treatment by this microbiome? How it's called, implantation? Or
yeah, so I haven't really come across many kind of studies that have shown success about fecal transplantation, right, as such in IBD, but I know that is definitely where it is going. As far as I know, there were some studies about also trying to capsulate that microbiome of a healthy individual in a capsule that you would give to a patient or rectally essentially administer that, which sort of improves, or let's say repairs, this gut dysbiosis that you would see in patients. So I think it's at a still very early but a very exciting stage, and I think as more and more of the microbiome landscape becomes clearer, it'll sort of give us an indication as to how the microbiome plays a role in terms of how a patient responds to treatments, in terms of prognosis, both in colorectal cancer and IBD as well.
Yeah, that's really interesting. And actually, from your lecture, so you presented in case of IBD the standard treatment protocols, and as you also mentioned, it seems to be it's working, but unfortunately not in all cases. Are there any new kind of drugs which are on the way, and we should hope for them? So
yeah, so I think at the recent sort of DDW and ECHO meetings as well, ustekinumab actually has, so again, I mean, the problem is that majority of the IBD drugs seems to be still focused quite significantly on TNF. So there is a bit of a difference in terms of treatment as well. So ustekinumab recently has now been approved. What we do know is there are certain small molecule inhibitors as well of certain kinases that are now kind of under phase 2 clinical trials that have shown certain degree of promise. So I think in the next couple of years we will start seeing improved second or third generation biologics that actually will be coming out, hopefully. But I think the problem is not just developing new treatments, but it's also being able to predict treatment progression at a much earlier stage, because these patients tend to come at the clinic at very sort of different rates. Some come at the very late stages, some come at the early stages. And at the minute, the only way to sort of predict disease progression is really doing endoscopy, with looking at the biopsies, looking at the histology. There are other markers like fecal calprotectin, say, for example, that is measured as well quite regularly. But again, none of them really give you an accurate prediction really when it comes to disease progression. So I think that's another sort of a challenge that needs to be overcome in the field of IBD.
Right. And what do you think these kind of studies, especially with the help of bioinformatics you presented with the studies which where you study like gene expression of the samples coming from the patients, can it help to predict the disease progression and essentially eventually maybe also treatment?
Yeah, so and that's exactly what we are trying to do in our own group, is that we have recently completed gene expression studies between progressors and non-progressors of ulcerative colitis. We have identified distinctive changes, distinctive gene expression patterns that are different between progressors and non-progressors. Also, what particular immune cell types are specifically different between both these groups, and we're doing the same thing with the methylation as well. So the point would be that if we're able to identify some sort of a biomarker, whether that be a tissue-based or a blood-based biomarker, ultimately that should hopefully allow us to kind of predict disease progression a bit better in the context of IBD.
Yeah, interesting. Okay. Yeah, let's wait for more questions. So we are still. So please write your questions in Q&A. So we are not in hurry. Just if you have any questions to Dr. Sudipto Das.
I think
Alexander, we have a question in chat. You can.
We have a question in chat.
Yeah,
okay.
Sorry.
All right. It's yeah. Right. Well, let me find it. Okay. So, do you observe a high degree of gene correlation or anti-correlation across the groups between the intermediate molecules of master regulators and so forth down to the signaling cascade?
That's a really interesting question. So the answer to that question is we haven't looked at that. So we haven't looked at the intermediate molecules and their kind of their downstream effectors really, and see if they actually correlate or not. But that is actually a really important point. I think we definitely can look at that, given the fact that it's within the same data set, to see if the gene expression correlation correlates with the intermediate molecules or not. But what we have done, obviously, which I did show, was that we have done this correlation when it comes to transcription factors and master regulators with gene expression. We haven't done that for the intermediate molecules. And one of the reasons I suppose we didn't do that was in each network, as you can imagine, there are a number of different intermediate molecules. It's not just one. So it's pinpointing down to that specific intermediate molecule is a bit tricky, no matter how smaller your network is. So I think that was a bit of a challenge, but that's something that definitely we can definitely look into further.
Right, to delineate the whole mechanism, right? To
Yeah.
understand how things going on. Right. Okay. Thank you for that questions. Yeah. So we got more questions in the chat. No problem. So, how are the master regulators manipulated during the disease progression?
So to be honest, I think that's quite an important question to answer in the sense of disease progression in general, and that's what we are trying to do. What we think is that these master regulators seems to be switched on. Let's say, for example, in the case of the IBD study that I showed, we think that these master regulators are being turned on once the patient is treated with the biologics, and ultimately, depending upon the master regulator that is being turned on, it is controlling a set of genes which ultimately impacts a patient to respond or not respond to treatment, and that's what we think is happening. Now, we are trying to look at in terms from an experimental point of view. As I mentioned, from the IBD perspective, again, is that we've identified these master regulators. So what we intend to now do is that we have in vitro kind of cell line based models where we will be knocking these master regulators down and looking at the impact of these master regulators on the intermediate molecules, on the transcription factors and their consequent genes, to try and firstly experimentally validate that network to make sure, yes, that is the case. But more importantly as well, that once the knockdown has been successful and we validated it, we would be looking at different sort of phenotypes, say for example, inflammation or metabolism per se, to see if there is an actual impact, for this master regulator as such in IBD, which hopefully will then rationalize, it as a potential therapeutic target or something that needs to be further investigated in vivo models even.
Very good. Interesting. Okay. Right. Until a new question is coming, let's come back to the first study you presented.
Yeah.
It's really exciting actually to see how different we are in the populations to respond to to disease mechanisms.
Yeah.
And in that study, so you presented this difference in immune actually response genes or immune involved genes.
Yeah.
And do you think it will lead also, or can lead to a difference in the treatment? For instance, using the very modern drugs which involve, especially in this case, checkpoint inhibitors, perhaps.
Yeah, so we don't really see PD-L1, PD-1, the standard checkpoint genes coming up, but we do see, key inflammatory molecules like IL-8, say for example, CXCL8, all of them where individual targeting agents are being developed. So I do definitely think that these master regulators that we've identified, which we think are driving this anti-tumor immunity, could potentially be targets of treatment. So that definitely would be. That was basically our kind of take-home message, apart from explaining what is underpinning or what is impacting this this phenotypic difference between both population of patients.
Right. Okay. And this can lead to kind of I wouldn't say the personalized therapy, but at least to prevalence to the certain types.
Yeah, at least between the two ancestry groups, definitely. I think that's what our study really showed, is that there is a clear-cut difference in terms of inflammatory profile between the two ancestry groups, and we are not the first people to show the immune differences, particularly because there are other people who've done immunohistochemistry on these patient groups as well, and have shown exactly the same kind of immune changes like IL-8 particularly, or CXCL8, both of them to be significantly lower in African ancestry patients compared to European ancestry patients with colorectal cancer. So that has already been shown. So I think our study really kind of reinforces what we have seen, what they have shown previously, but more importantly, also reinforces that you could look at these patients differently from an immune perspective, but what is driving these changes, and could the drivers then ultimately become important therapeutic targets?
Right. Is it specific for cancer, or can lead also differences in other types of immune-related diseases?
So we don't know. That's the biggest question. We were only focused on colorectal cancer. However, one of the studies that was published, I think it was last year, again focused on cancer, but that was the largest TCGA study where they looked at they did multi-omics, but they looked at all the cancer types within TCGA, looking at different ancestry populations. They found very similar things as well. we are in the process of doing a similar sort of ancestry-based analysis in IBD as well, looking at differences between European ancestry patients and Asian ancestry patients, actually. But in this instance, we are primarily looking at mutational differences as opposed to transcriptional differences. So that's underway at the minute for us. But as far as I know, and my guess would be that this, looking at it as from a cancer type is just the tip of the iceberg. I would presume that such differences also prevail in other disease types as well, particularly the ones that are driven through autoimmune processes.
Yeah. It's very interesting. Okay, so I guess we got some more questions. Yes, there is one question. How can we know these masters or master regulators are special to a cancer type, not expressed in normal tissues?
Yeah, so the gene expression analysis, so this kind of brings me back to the gene expression analysis that we did from where we identified the master regulators. So the differential gene expression analysis that we had done for the gene expression studies, we had made sure that the gene expression was only accounting for tumor specific changes. So everything that was normal was subtracted from it. So if you can imagine, we started with the tumor specific gene expressions, and then we derived the master regulators from there. Now, what you were saying is very true. IL-8, granzyme B, CXCL8, RANTase, all of them are key inflammatory molecules that not only control, they're not only controlling gene expression in cancer, but they can be, they can be specific to other cancer types or even go beyond cancer. So these master regulators are not specifically cancer associated as such. They are known inflammatory molecules that tend to drive common regulatory, immune regulatory processes. We, however, have identified this through transcriptional networks which are tumor specific. So for us, these seem to be driving the case, but in other kind of disease types, they very much could be driving similar changes as well.
Yes, very interesting. Okay, thank you for answers. Yes, we are still waiting for more questions. Let's see. Yeah, generally, I think this all topics of personalized medicine is really really interesting right now. And for us, as you understand, it's very important to figure out how these bioinformatics approaches can help in that direction. And in your lectures, in your lecture, you clearly showed it in three different studies. They kind of related to each other, but still different. So what do you think? What is the next step?
I think the next step really is like the discovery is happening. I think the next step is really to kind of get down to the detail of how these master regulators function, particularly treat them as individual therapeutic targets, but more importantly, these master regulators, you could go back to data sets and see, are they up or down, and could they then also be utilized as companion diagnostics, in terms of identifying which patient should be selected for the treatment, which patient shouldn't. So I think these kind of transcriptional networks really are multifunctional in that not only it gives you a really clear picture in terms of mechanisms, how diseases work, how patients respond to treatment, but also you do identify in parallel novel ways of looking at gene expression data as well. What is controlling, because a lot of the times when we look at gene regulation, it's either genetic or epigenetic factors that we know control it. So I think master regulator really is another important layer of complexity that seems to control gene expression. So really I suppose that's what we have started to do in the last couple of years, is we don't look at, individual regulatory aspects. We tend to take all of them together. So any study that we are doing, we are looking at the genetic factors, the epigenetic factors, and also master regulators, which all in tandem might ultimately control gene expression. So I think that's where I would see this going next. But yeah, absolutely. I think the master regulators really do have a significant utility, both as a biomarker and as a potential novel therapeutic target.
Perfect. That's what I wanted to hear. Thank you very much, dear Sudipto. And right, so with that, we can finish our webinar today. And if you still have questions, you have here several thank you people sending to you. And you stay with us, so there will be more webinars.
I think there is one question there. Are you taking?
Okay, yes, so we got one more question. Very good. Yes. Are you talking polymorphism into? Are you taking polymorphism into consideration for the differential gene expression or master regulators in your ancestry analysis?
Yeah, so we did take polymorphism into consideration. We did make sure that the gene expression changes that we were observing was independent of polymorphisms or any genetic changes as such. There were no mutations within them. We also looked at polymorphism within the genes itself. So again, because obviously this is from Cancer Genome Atlas, we knew whether these genes that we were looking at, had polymorphism in general or not. So we did take that into account.
Mm-hmm. Yes, but that's, I guess, another level of complexity.
Yes, absolutely, absolutely.
Yeah, in future probably we should sequence the whole genome of the patient, but that's to see.
I mean, it's getting there, to be honest. A lot of like we look at the 100,000 genome project that has now been published in Science.
Right.
They have done whole genome sequencing, a whole exome of majority of the patients that they were looking at. So I think that's the way really to go forward.
Exactly, it will come. Okay, I think with that we can finish, and thank you very much again.
Thanks a million.
And, yeah, stay with us, and we wish you all the success in your future work. And yeah, we'll talk more in future.
Absolutely.
Thank you.
Absolutely. Thanks very much, Alexander, and thanks everyone for your attention.
Goodbye.
Goodbye. Thank you.